Hi, I'm Shella Keilholz talking about the intrinsic functional organization of brain activity. I doubt I need to convince anyone here that the intrinsic activity of the brain is important, but if you do need a little bit of persuasion, if you look at the energy budget, of course we know that about 20% of the body's energy goes to the brain, and of that 20%, only a small sliver goes to activity associated with processing of stimuli and cognition. Most of the rest of it is devoted to this sort of background intrinsic activity. So we know that it's important. We also know that intrinsic activity is influential. If you look at the response to activation to the same stimulus given at different phases of the brain activity, you can see that you get a different response. So it matters what phase this background activity is in when you're trying to do a task or looking at the response to a stimulus. And also, this activity is persistent. So we tend to think of resting state fMRI as something that you do to look at cognition or cognitively relevant information during wakefulness, but the same sort of networks persist during sleep, during anesthesia, across species. So they seem to be some sort of fundamental mechanism that organizes the brain's activity. Brain activity occurs at a wide range of spatial and temporal scales. There are things of interest that happen at a single synapse over the course of milliseconds. Of course, we can't see those with fMRI. Where we can start to look at with fMRI are things like these mesoscale organizations like columns and layers. So maybe you're interested in the inputs and outputs at different layers of the cortex. Maybe we're interested in cortical columns where a single column might correspond to a single whisker in a rat or a mouse. So going then up to kind of the next level, there are these thalamocortical circuits and other circuits that involve, say, cortical areas and deep brain nuclei that have been studied for years using techniques like electrophysiology. And these are kind of wider spread than the mesoscale organization and coordinate activity across areas. Taking a larger view still, there are nuclei, especially deep in the brain, like the locus coeruleus or the basal forebrain, that have very widespread projections and are involved in arousal and regulation of cortical activity. So they may have very wide-ranging influences on what we see in the brain. We've been talking about neural activity so far, but there are other players in the game, and I think astrocytes may be particularly important. Astrocytic activity has been tied to neuronal activity, but it's also tied to hemodynamics, making astrocytes, very well positioned to kind of translate what we see at the neural level into what we might see with resting state fMRI. So all this activity at all of these different scales is mashed together into what we observe with the resting state fMRI signal. And that makes our challenge to take that signal and somehow extract out of it interesting things about the neural activity in the brain, particularly its intrinsic structure, where we don't know the timing, we don't know the stimulus, and we have, to come up with other ways to characterize what's going on. There are a lot of ways to look at resting state fMRI. You could argue that there are too many ways. They're all valid, they're all useful, and they all provide really interesting information about the data. But each of them kind of has, a different focus. So I'm going to group them just kind of loosely into two categories. One that detects time-locked patterns of activity across regions, where it picks up things that occur in sync across different areas, and other analysis methods that also pick up time-lagged activity, where you can see activity that may follow activity in one area then happen in another area. So if all of these analysis methods are valid, why does it matter which one I pick? And to me, the answer is that when you make an analysis choice, you're kind of automatically assuming a model for what's going on. And let me use a couple of examples just to show you what I mean by that. So when we talk about seed-based correlation, we've got an area and we're looking at other areas where activity goes up and down in sync with that area. And at least for myself, I tend to think of that as, being something that continues over time. These two areas are always talking to each other, always communicating, always coordinating their activity. Whereas if we talk about coactivation patterns, we think of patterns of activity that recur over the course of the scan and maybe don't last very long, maybe just for, a second or two. So these are kind of states that the brain might pass through, and they don't imply that same sort of lasting relationship that maybe you think of when you think of correlation. So if different analysis methods imply different models and different choices, then we can ask, what is the right way to look at resting state fMRI? And I put right in quotes because I don't mean to imply that the other ways are wrong, but I do think that we can aim to get closer to the underlying features of the neural activity to find a model that better describes what's actually going on. And I was trained as a physicist, so when I think about models, I like to think about some of my favorite physicists and the elegant models that they come up with. And when physicists say that a model is elegant, usually we mean that it's simple. You want to capture a lot of information with just a few components. You want something that's based on the signal itself, ideally, not some secondary measures. Whatever model we decide on should explain everything that we find about resting state fMRI. And for intrinsic activity in particular, we know that this model has got to have some sort of dynamic aspect, right? Because things should change over time. So our current favorite model are quasi-periodic patterns, or QPPs. Now we've also called these complex PCs, we've called them spatiotemporal patterns. The name doesn't matter. We're always talking about spatial patterns of activity that evolve over time and reoccur in the same sort of configuration at multiple points in the scan. And so when we run one of these analysis techniques, what we get out are some sort of template that describes the spatiotemporal evolution of the signal in one pattern, and then we get some kind of time course or weight that tells us how strong that particular pattern is at any given point in the scan. So I'm going to play one of the templates here so you can see what it looks like. This is from data from the Human Connectome project. Warm colors indicate activity above the mean, cool colors are activity below the mean, and you see that this pattern kind of divides the brain into these two anti-correlated networks, which we've seen in other analysis methods, and you capture the alternation between these networks and the propagation along the cortex that occurs as the networks switch. Now what you don't see when I play this is that this pattern also involves subcortical structures in the cerebellum, so it truly is a whole brain pattern. You can find multiple QPPs, or multiple spatiotemporal patterns, in our data. We found that about 3 is usually a good number for human data, and it explains most of the features that we see. So, to examine exactly how things like QPPs relate to something more standard like functional connectivity, what we can do is regress those spatiotemporal patterns out of the data and look at functional connectivity before and after we do that. So over here, what we have is the functional connectivity matrix, again for Human Connectome project data, before we've done any QPP regression. And then this lower half of the triangle is that same data after we've regressed out the 3 primary QPPs. And you can see just visually that we've removed most of the functional connectivity that was originally in the data. So looking then at other features that people have extracted from resting state fMRI, first we noticed that the QPP at one time point looked a lot like the functional connectivity gradient, and that if you watch the evolution of the signal in that QPP, it would sweep across the functional connectivity gradient. And then Taylor Bolt did this really beautiful implementation of a spatiotemporal pattern finding algorithm with complex PCA, and an absolutely exhaustive comparison of the components you get out of that to different analysis methods like coactivation patterns, hidden Markov models, eigenmaps, seed-based correlation, ICA variations, all sorts of stuff. And everything that he tried, we could pretty much explain with about 3 complex components. So this really implies that those 3 QPPs, or those 3 complex PCs, account for what we're seeing in the data, these spatiotemporal patterns. So if we go back to our original, analysis models that we were talking about, if we think about seed-based correlation now, maybe we interpret them in the light of these spatiotemporal patterns. And instead of thinking of areas as necessarily being a functional network kind of in isolation, these are areas that activate at the same phase of the QPP. Similarly, for coactivation patterns, maybe instead of being these recurring instantaneous configurations, these are snapshots of a phase of a QPP. So realizing that these spatiotemporal patterns account for most of what we see in resting state fMRI just gave rise to a bigger question, because we still don't understand what organizes these large-scale patterns and what they reflect. So we came up with a list of possibilities, cognition of some type, noise of some type, neural activity of some type, and we ruled out the cognition really quickly because these patterns are relatively periodic, they recur over the course of the scan, and we're not having the same thought or doing the same task unconsciously in that same sort of manner. We also looked at noise from motion and respiration, and it took us quite a while, but we also ruled that out, looking at a bunch of different data sets where those were very highly controlled. They don't appear to give rise to these kind of constant spatiotemporal patterns that we see, which leaves us with the idea that these are probably neural in origin, and maybe they reflect different sources. We came up with a couple of ideas. One is that you have this wide-ranging input from those neuromodulatory nuclei, so maybe this is related to that pattern of input. And another was that you have this network structure of the brain, and maybe there's some sort of resonance of that network structure that helps to shape the intrinsic activity. We've done a lot of work that I'm not going to get into in this talk to show that these QPPs are tied to neural activity. But just as one quick example, we've looked at QPPs in rats that were sedated or where they had been given a dose of sodium pentobarbital to make them basically flat electrically, so no neural activity, but they're still ventilated, their heart's still beating, so that sort of noise should still be there. And what we found is that for each of these, the top row is from the sedated rats and the bottom row is from the isoelectric rats. We found that in the isoelectric rats, the QPPs were very disrupted, didn't have much spatial organization, and didn't appear to really represent anything physiological, again supporting that there is a neural source for these QPPs. So we've begun to examine the possibility that some deep brain nuclei are involved in the organization of these patterns. We focused on the locus coeruleus in particular for a few reasons, the most important of which is probably that we have a collaborator with a lot of expertise in that area who could help us. So first we gave some rats a neurotoxin that's specific to the LC, and we found that that weakened the QPPs to the point where they were undetectable. So that was encouraging. With that under our belts, we went on and did some optogenetic stimulation of the LC. We're still analyzing this data, but what we've seen so far is that stimulation increases functional connectivity in the rats that have the channel 2 rhodopsin, which makes sense. And our very latest just this week analysis says that stimulation of any type of the LC also increases the strength of the QPP. So I think we're gathering evidence that it's true that the LC is involved in this. That doesn't rule out the involvement of other nuclei. And I certainly think that there may be kind of the whole neuromodulatory system that's involved in the generation of these patterns. The other possibility we wanted to examine was that these might be a resonance of the network structure. And so we built a brain network model where you divide the brain into different nodes. You use the white matter between those nodes to give you kind of some structural weight to the connections. And then at each node, you can use a neural mass model to describe the local neural activity. And then you let the different nodes interact over the structural network, and you can produce some sort of synthetic signal that would be kind of a surrogate for resting state fMRI. And so we took that signal and we put it through QPP analysis. So I'm showing the QPPs in a little different way here. So in these plots, each row is a parcel in the brain, and you're looking at the activity in that parcel over the course of the QPP template, with yellow being high and blue being below average. And so this one here at the top left is what we get from actual resting state fMRI data. We looked at two different models, a firing rate model and a Kuramoto model, here and here. And you can see in each of these models, we've got kind of this bimodal flip between two networks, activating and deactivating. But we don't have any of the complex propagation that we see in those QPPs from the actual data. So I think that there is something to this network resonance idea, and maybe it's limited because a lot of our network models right now don't incorporate physical adjacency, which may be important for this type of analysis. So to wrap this up, we've become convinced that there are three large-scale spatiotemporal patterns that account for most of what we see with any other analysis method for resting state fMRI, and that they arise from some combination of neuromodulatory input and network resonance. I think this implies that we really need to think about how to interpret resting state fMRI. What does it mean if we see a change in functional connectivity in patients with depression, for example? How do we make sense of that? But at the same time, I think there's an opportunity here for us to come up with new theories about the systems level organization of the brain and how we might manipulate it to treat clinical disorders. The other thing that I want to point out is that while these patterns account for about 70% of the functional connectivity and the structure we see in resting state fMRI, they only account for about 1/3 of the variance, which means that 2/3 of that is still unaccounted for. Now, we know that, from fMRI, we know that there are small localized changes related to differences in cognition and task performance. Those must still be in there. I don't know if we can get them out, but maybe if we can extract some of the systems level signal, we can get to those more localized changes that might be interested. On the other hand, fMRI is very noisy, and we know there's a lot of noise in the signal. Maybe that's going to swamp anything of interest. I think we will just have to see. I just want to finish by pointing everyone to some resources that we have available if you're interested in trying this type of analysis, and to acknowledge everyone who works in the lab and our funding. And thank you all very much for listening.